Hi guys, welcome back. So I recently made a macromolecules video lesson, and here are just some past USABO questions to go with it. So for each of these, please pause the video and try to do the question by yourself, and then I'll go over it. Okay, so for this one, it's asking which two amino acids are least likely to occur in the middle of an alpha helical protein structure. So if you remember, alpha helices, they have two major amino acids that they don't like, and that's proline and glycine. So the reason glycine isn't good for alpha helices is that it's simply too small. Its side chain is just a hydrogen, and that really messes with the structure. As for proline, proline is an amino acid, and overall the shape of it just isn't compatible with an alpha helix. So the answer is B. All right, now try to do this question from 2018 opens. All right, which of the following amino acid substitutions is least likely to be disruptive to protein function? So for these types of questions, we have to look at whether the two amino acids listed are similar or dissimilar. So it's asking for least likely, so we want to find the pair of amino acids that's the most similar. So that could be in terms of like charge or polarity, right? So if we look at lysine to glycine, so lysine is basic and glycine is nonpolar, so that's a very big change. We'll go to B. Cysteine to leucine. Cysteine is polar and leucine is nonpolar. So again, a pretty big change. Then we have alanine to proline. So I believe both of these are nonpolar, but proline has some issues with alpha helices, as we just mentioned. So we'll keep C in the back of our mind, but let's move on and look at the other choices. D is aspartate, which is aspartic acid, to valine. So obviously aspartate is acidic and valine is not, so that's a big change. And then finally we have alanine to valine. And this is our best choice because they're both nonpolar. So like alanine and proline are also both nonpolar, like the C pair, but proline disrupts alpha helices while valine doesn't, so our best choice is E. Alright, so now another one from 2018 opens. This question, we're going to do it in a way that's pretty similar to the previous one. We're going to look and try to find the amino acid sequence that's the most different from the one they gave us. So starting with the first amino acid, A has alanine, the other ones have valine. So alanine and valine, they're both nonpolar, so that's not going to make a big difference. And the next difference is right here in the fourth amino acid spot. And B has glutamine instead of N, and that's not going to be a big deal since both of them are polar. And then we have D having Y instead of A. So tyrosine is polar while A is nonpolar. So we have D as a candidate right now, but we'll look at the rest of the sequence. Again, E has I instead of L. That's not going to be a big deal. They're both nonpolar. And then A versus G, again, they're both nonpolar. So that gives us D as the most different sequence. So D will be the answer. All right, take a look at this one. This one's from 2015 opened, and it's about DNA. Okay, so I don't think I actually talked about this in the video lesson, but if you look at the pairing, when adenine pairs with thymine, there are 2 hydrogen bonds, and when guanine pairs with cytosine, there are 3 hydrogen bonds. So what that tells you is that when G and C pair, they have a stronger bond in between them. So if you're in a situation where your DNA can denature very easily, in this case because of temperature, you're going to want DNA that has stronger bonds. So that's going to have more GC base pairs, which tells us the answer is B. All right, look at this one from 2014 opens. Okay, so it asks what is not a component of the cell wall or cell membrane. So if we look at our first choice here, A, this is a phospholipid. So this is kind of like the choline group. This is the phosphate head. And then this is like where the glycerol is. So basically this is a phospholipid, and that is a part of cell membranes. So it's not A. If we look at B, it's chitin. You can tell because sugars are going to look kind of like this. This is what's called like chair form in organic chemistry, and it's basically just another way to represent a ring. So we're used to seeing kind of like the six-membered ring for, like glucose and other sugars, right? But this is just another way to represent it. And we see that there's a nitrogen in this part of the sugar, so we know this is chitin. And obviously chitins are part of fungal cell wall, so it can't be B. If we look at C, C is cellulose. You kind of just have to know for these two, but it shouldn't be that hard to deduce. Like, generally, like for USABO, they're not going to expect you to memorize like very, very obscure, biochemistry. So it's pretty safe to assume, oh, this is probably cellulose, because you see that like this is flipped compared to this, and then that kind of tells us it's like the beta glucose that has flipped bonds. So yeah, and then this one has nitrogen on the side. And then D, it's a serine. You don't necessarily need to know that. You can just say, oh, I don't know what that is. That looks kind of strange, and then set it aside. But E, it's cholesterol. It's a steroid. Again, like, other steroids, they look different than cholesterol, and it could possibly be one of them. But usually on like a USABO test, you're not going to find something super obscure like that, so it's safe to assume this is cholesterol. Cholesterol is found in cell membranes. So yeah, it's going to be D, which is serine. An amino acid, you're not going to find a lot of that in your cell wall or cell membrane. All right, and then last question. Okay, so we have a substitution mutation that goes from a glutamic acid to a lysine, and that's from an acidic to a basic amino acid. So let's look at all of our answer choices. This is from a semifinals test, and it's part B. So that means we're going to have to select multiple choices, and we only get it correct if we find all the answer choices. So let's look at A. The change from E to K would be expected to increase the protein's net negative charge at a neutral pH. So we went from an acidic amino acid to a basic one. Acidic amino acids are negatively charged, and basic ones will be positively charged. So no, that's not true. B, the change from E to K could potentially impact ionic interactions in the tertiary structure of the prion protein. Yeah, because we just swapped something negatively charged for something positively charged, so it's reasonable to assume that. Now let's look at C. The E to K change is unlikely to impact the primary structure of the prion protein. Well, that's not true, because primary structure is your string of amino acids, and if you're changing your amino acids, well, that's going to affect the primary structure. Now to D, the protein change is a result of a G to A change in the mRNA sequence of individuals with the disease-causing allele. So if you go into your chart and you'll look for glutamic acid and lysine, you do see that's the case. That's just a case of reading the chart. If you don't know how to, I believe there's a video up, one of the genetics videos, the Central Dogma one, I think, that tells you how to look at these. So yeah. And then let's move on to E. The protein change may have been a result of a nonsense mutation in the original DNA sequence. So again, for this one, you have to have some outside kind of like genetics knowledge, but a nonsense mutation results in a stop codon, which is one of these three, and that's not true. So our answer is going to be B and D. All right, thank you so much for watching. I really hope this was helpful in helping you prepare for your competitions. If it was, please leave a comment or a like, and see you next time.